the 12th of our Fireside chats. We're recording today's chat as we usually do, and we've posted all the previous 11 Fireside chat recordings to Coursera. And you can find those. You can find those in the resources tab of your course. And this one will get posted also shortly after the conclusion of today's chat. Just a couple of reminders, and maybe if you haven't been here before, things to keep in mind. The microphones are muted, videos are turned off, and please don't turn them on. We do have a guest today, and we're going to have a question and answer session after her presentation. But if you want to submit a question to either me or to her, please do that using the Zoom function, the chat function in Zoom. You don't need to wait until the Q&A part of the Fireside chat starts. You can do that anytime. And when we get to the end of her presentation, then we'll take the questions in the order in which we've received them. And we already have a few that have come in advance. If your question doesn't get answered during today's Fireside chat, we will get to it. We will respond to it, to your questions. And that will get posted in Coursera for those of you that are enrolled in the course. So our guest today is Dr. Kelci Miclaus, and I am really pleased to have her here. She's Senior Director of Veeva Stats, and there she leads product development and strategy for Veeva Systems, statistical computing and environmental solutions. She previously was at JUMP, where she helped create, develop, and manage statistical software solutions as Advanced Analytics Senior Manager for JUMP in the Life Sciences Division, rather, at SAS Institute. Kelci has a PhD in statistics from North Carolina State University with emphasis in biomedical and genomic statistics. She is a subject matter expert in the areas of software, data platforms, analytics, data visualization, and advancing and operationalizing discoveries with genomic, translational, and clinical data life cycles in the life science domain. Really pleased to have her here today. She's really an expert in this area, and she's going to talk with us about clinical trial design in this sort of modern era that we're in of real drugs, big data, lots of interesting things going on. So Kelci, thank you very much for coming to participate in today's Fireside chat, and I'm really anxious to hear everything you have to say.
Thank you so much, Doug. I'm happy to be here and happy to talk on this subject, as it's definitely one close to my heart, and now one that much more people are aware of given the current climate around clinical trial research, given the COVID-19 pandemic, of course. So let me go ahead and share my screens and get into that presentation. As Doug alluded to, the focus of my talk is really on the modernization of clinical trial design as we come up with more complex data sources and complex therapies in modern medicine, as well as with modern technology. There's going to be a clear focus here on not just the design principles or the impacts, but on the statistical methodology consideration, given how these designs have evolved over the years as therapies and data and technologies evolve. Typical disclaimers, these are my thoughts, not necessarily what I'm doing in my day job. I also should be very frank that I am not a DOE expert, so much of the framework of my presentation is the impact of different designs and how clinical trials are designed on the statistical methodologies in order to efficiently use that data to produce safe and effective therapies. Quick overview of what I'll present today. Largely, this is going to be a high-level presentation of the aspects of clinical trial design, but clinical trials is an enormous field within life sciences as another enormous field. And there's multiple phases where you have different design considerations. So I'm going to narrow our focus and our scope within that in the phase 3 clinical trial design, which I'll get to as I go through those. And really talk about that with the impact of how those design paradigms change or impact how data flows and information flows and how data is analyzed in the operational process of that clinical trial. An important consideration to always take into account when considering trial design in clinical trials is the regulatory guidance. So oversight from regulatory bodies, FDA, US, PMDA, CFDA, global regulatory agencies, and how they provide that guidance. So I'll key in on some of the key parts of how the guidance has influenced design, but also how the industry influences that guidance. And most of this will be in this idea of eras or the evolution of modern medicine and modern design and clinical trials, from what I'm calling big drug, real drug, big data, real data, and then of course the pandemic curveball. So let's get into it. So clinical trial phases, for anyone who's no longer familiar, I think in the past year now, many more people are familiar with how these are executed. But at each stage of this, there are very critical design decisions made. So if you think of phase 1 through 3, as shown on this slide here, this is a highly involved process that typically takes 7 to 10 years. So, you know, the money, the time, the effort that goes into these trials is not minimal. And so how to design these trials well and effectively to speed up that process or to make that process more effective is always what's driving innovation in clinical trial design. If we think of those first 3 phases alone, outside of preclinical or post-clinical research, phase 1 is typically, you know, toxicity analysis, dose finding out what's that maximum tolerable dose. So it's very small sample sizes set on making sure we have something that is effective, that we can actually treat, or not effective, that is not toxic to the level that it will only do harm. Phase 2 is when we start to get a little larger, we start to consider trial designs, but it's still really just this early design play of trying to make sure before we launch into a larger trial that it's going to show some measure of efficacy and going to show some measure of safety. And then phase 3 is where we are going to focus today around much of the trial design aspects. Within this process, phase 3 is the one that takes the longest and the most resources, and of course has the most people. If we're talking of phase 3, and in a classical sense, you can think of the data as it flows through a clinical trial system of you've designed a clinical trial and then you operationalize that. You start up that trial. Typically, that means you have multiple sites. Almost all trials now are no longer a single site as we globalize clinical trials. So we have possibly multiple centers, multiple sites around the world, and data flows in through that with electronic data capture into a central repository for data management. That data management then flows into analysis-ready data. This is where I focus most of my career in, is this statistical analysis component. And it's at that point when you really start to be able to see signals of safety or efficacy, and that's what's going to drive critical decisions of the trajectory of that trial. And of course, it's also going to end up with a final statistical analysis that is what's submitted to the regulatory agencies for drug approval. As I'd mentioned, guidance and regulatory oversight is critical to this area. And so, you know, this is probably the unifying document, the guidance, this E9 statistical principles for clinical trials around good clinical practice on how to execute, design and execute and analyze clinical trials. Much of this was done at a very early stage when most pharmaceutical companies were really driven towards big drugs. and simple. So we wanted to keep things as simple as possible so that we can show at its basic level, the drug works and it won't hurt us that badly compared to how well it works. So this guidance, of course, has driven a lot of the design, but a good thing to keep in mind with guidances, and as I move into the next ones, much of these guidances are really driven by how the industry changes, and then guidances are released to approve that process. So, but jumping into that, from the original guidance around the execution and design of clinical trials, the gold standard had always been to consider a traditional, very simple, randomized clinical trial. This was always in the effort of finding that blockbuster drug, finding that drug that's going to work for a broad population and is simple therapies. So because of the good clinical practice considerations, there's a huge ethical burden on designing these simple trials to make sure that there is enough patients to show that there is a clinically meaningful effect, but you don't want to expose too many if there is actually a risk or an unnecessary risk in the development of this new therapy. And so with these traditional randomized clinical trials, every set of that analysis needed to be set up prospectively in the study design and the trial design, predetermined, completely defined, measurable endpoints, and then power and sample size calculations in order to reach those endpoints were based on that. And it was a go or no go situation of, if you don't meet that endpoint, the trial fails. It goes even to the level of, much of the statistical considerations around that are how you handle intent to treat. Natural things happen. Someone has to go off treatment. They have a bad reaction. But in the trial design, this rigidity means that you actually still treat them as if they're being treated, because that could upend the entire prospective design. As I mentioned before, usually this is a multi-center trial, so it's all around minimizing bias and keeping patients safe. So it's traditionally just a simple randomized block design. In order to meet the criteria for minimizing bias, oftentimes there's very, very clear protocol deviations set in place for inclusion-exclusion criteria of who can enter a clinical trial. And then there's also great operational oversight on these trials, which is a design aspect in terms of quality design for which sites to pick of its own that I'm not going to discuss here. I'm going to go through this quick, but the impacts of a simple design are simple statistics, which is great, right? So we have a very simple way of looking at that data. We have checkpoints for efficacy, but it's really nothing that happens from those checkpoints except the data looks good. We haven't had any signals that mean we have to stop. Let's keep going. And then we can make a submission. That's not really the real world. So big drugs are mostly discovered. I mean, most of what we take now are complex therapies that serve a purpose specifically for us. So what has now been released, this actually just got released this month. Of course, it's been a work in progress for several years. This is a clear place where industry has now driven guidance that big drugs in a vacuum do not reflect the real world. And this addendum to that statistical principles really focuses around this concept of an estimate, which instead of saying you have this fixed rigid estimate that you have to hit, this effect size you have to hit, you're really incorporating all of the factors that can hit you in the development of a complex therapy, who the right population is, what the efficacy endpoints will actually be in point of. And the big one here is this idea of intercurrent event handling. Before we use safety checkpoints just to say the trial's proceeding as planned. But now there's design aspects that allow us to build intercurrent event handling into how the trial design changes as we randomize and collect more patients. And that's what's really led to the next 4 eras of modern clinical trial design, adaptive clinical trials. This is no longer a modern term. This is just the standard. Calling it traditional is a little strange because now most trials that we encounter are adaptive clinical trials. And the reason being that they're flexible, they're more ethical for patients. You get to make decisions as you go through versus the rigid start point. And it, of course, brings efficiency. So you'd think of a traditional as the design, contact, analyze. That's all you can do. But the idea to be able to review and adapt the conduct is where that's changed. And of course, you can even think of review and then adapt the way the design works. One clear thing to mention, though, is as we enter into adaptive trials they still do mean that upfront we are defining that adaptability. So the highlighting example, especially right now, is oncology. Drugs are complex. You can't really put them on a control. There's serious considerations with survival to keep in mind. So a lot of the new modern designs have been driven by the field of oncology. I'm listing here just a couple of the types of designs where you really bring in that interim analysis, not just as checkpoints, but as decision points of how the trial changes, how the trial proceeds. So group sequential can be how you actually rechange the groups or resize the sample size. You no longer have to think of this fixed sample size. You hit an efficacy endpoint and you can decide at that stage, can we enroll more patients? Multi-arm, multi-stages, you handle crossover designs, you handle this idea of people on multiple therapeutics, some of which might be a standard of care, some of which may be placebo, or some of them might be combination therapies that each have their own types of treatment effects. Adaptive randomization is another type where you can actually, as you learn about the populations, you change the direction of how you randomize patients into these different arms. And of course, Bayesian approaches are love in this field, as it makes sense that as you learn information, you incorporate that into the design. Another interesting area that's really come out of this too is benefit risk. This is kind of different from the others where it's now you're balancing not just safety and efficacy, but cost of clinical trials. What many pharmaceutical companies have realized is you can spend 10 years on a drug that's just not going to bring what it needs to bring. So benefit risk is this idea of for what the drug can do, can we really afford the cost of this trial, of this development? For better or worse, that's now a consideration people put into the design of clinical trials. Just a quick note here to the parts where I find a lot of value and interest in my field is how these designs introduce statistical complexities. And sometimes it's more complexities to the statistical analysis. And sometimes in this case, we're now having to produce efficacy measures, proof that the drug works or that it's safe at much earlier stages of clinical trials, which don't have the power of the traditional statistical methods. So we're moving to innovative types of early efficacy metrics, or even visualization, which is another area I'm really, that's close to my heart, even visualization to really provide that evidence that you can move to that next phase. As we go from the second to the third, we're really still in the same era of adaptive trials, but now we're entering outside of just big and real drugs into big data as we develop real drugs. As technologies got better and better, pharmaceutical companies, sponsors realized you can actually collect biological samples on entry to a clinical trial and make informative decisions on the direction of your trial from that data. So, genotyping technologies, sequencing technologies have drastically gone down in cost, that it is, it's something that can be done even in large trials. And this is incredibly important to adaptive trials, right? So we had to design them up front with adaptivity in them. Well, the best way to do that is to have the knowledge of how we might have to adapt. So they want to drive that design. They want to drive the understanding of should we enroll more patients and which types of patients. by using the biological pathways or biomarker discoveries that you can make based on the chemical compounds of a therapy, for example. And so really this is just a directed way to do better adaptive trials. And you can think of biomarker discovery typically as either prognostic, prescriptive, like determining, given a patient, should they take this treatment? And that's more of a hospital setting or a doctor setting. Or predictive. Can we actually take a patient and predict their outcome to a treatment given their genomic profiles? And this is an area where already there's been a huge increase in the number of biomarkers that are popping up in drug labels. Oncology, again, is a great example. We now know, hey, if you have this gene measurement, you shouldn't take this drug, or vice versa. So really what we want to do with this information is incorporate that signal back into the design. So two of the ways that, in my experience, that I've worked around methodology for this is in subgroup analysis and also in optimal treatment regimes. Subgroup analysis is a really key component to clinical trials. We always collect demography information, different types of information, and it's not always just the population as a whole. We have to look for signals where perhaps there's a safety effect that can be explained by a certain demographic population or a certain population that's categorized in some other way. And what subgroup analysis does is, especially if you use it with biological signatures, it looks for that interactive effect. It looks for the groups that behave differently. In fact, this was actually taken from market research called uplift modeling. And it's based on marketing of, hey, if I send you an ad in the mail, are you going to be more likely to buy my product, or is it going to piss you off and you're not going to buy my product? It's the same idea of, if I give you a drug, are you going to get worse from that? And if so, what characterizes the population that gets worse? Or is that drug going to help you? And what characterizes those that are benefiting from a drug? Another interesting area outside of subgroup, which really is thought of, given a drug, how can we find the right patients to treat it? Optimal treatment regimes is coming at it from a very different way of, how can we find the best drug for a given patient? So given a patient, what's the best drug that they should go on? And this is actually a unique aspect of clinical trial design, where now you actually can design the trial with some ethical considerations in mind. Knowing what I know about that patient, how can I make sure that they are randomized to where they'll have the best outcome and my trial will have the best outcome? And so you can actually do this with propensity score modeling and predictive modeling in order to do reinforcement learning mechanisms to design how that clinical trial should be randomized and which patients should go into each arm in order to optimize the treatment effect. So those were the two around the big data. But now we also have real data. So genomics, of course, is still real data. What do I mean here? Well, really, it means real world data. We've gone past this idea of clinical trials have to happen in a clinical setting in a super controlled manner, randomized controlled trials. That is no longer many of the purview of clinical trials as we consider how we take in all of the data, which is still big, of course. And I don't typically think even of genomics as just big. I think of big and real as together. What makes data big is complexity. And we're hitting that now. We're having complexity of sources of data, the structure of the data, the variation in the data, how you analyze that data. And now we're at that point where we're trying to incorporate healthcare data into the conduct of clinical research. This is a new area called pragmatic trial design. So this idea that you're allowing health systems or even doctor visits outside of a clinical setting to drive clinical trials. And one of the biggest benefits of this is diverse representative study populations. The inclusion-exclusion criteria can become so extreme to the point that we might have a drug that's efficacious, but is it actually effective when used by real people in the real world? This table, of course, I have the citation up here. There's a lot of differences. It's really just upending the entire original premise of a clinical trial design. Typically, we thought super simple, super homogeneous, clear randomization, double-blinded. I didn't even mention that earlier, but that's the gold standard. It's double-blinded. The patient doesn't know what treatment you're getting, and the person, the investigator, doesn't know what treatment that patient got to minimize bias. That's going away now, and we're considering new mechanisms to handle that. In terms of the complexities, though, it really does come down to diversity, asymmetry, and latency. All these different data sources that now have to be collated before we can even get to an analysis endpoint, to get to some type of insight if the drug is working. And for ethical considerations, we need to get to that point very quickly. Asymmetry is an interesting one too, especially with regard to missing data. So if we're working in a real world setting, well, first let's think of we're in a clinical trial. If you're in a clinical trial, very structured clinic visits, you have to go in, they take your labs, you say, I feel great, and you go home. If you're in a doctor's environment or home healthcare environment, if you feel good, you don't go to the doctor. You don't submit anything. You don't have any complaints, right? So there's this real key difference in the analysis of clinical trials with whether you're thinking of data that's missing or just a gap in the data due to the structural design. And there's also information latency. This goes both ways. So we used to get this through those regular doctor visits into the electronic data capture. And now we're thinking of healthcare systems where we actually, we're thinking of healthcare systems where you're trolling medical records or even claims. It could be medical claims. On the other hand, it could also be new types of data that come in much quicker. So you have to handle how you collate that information, the timing of it, and the differences in that information in order to get you to that point where you can make a call on efficacy or safety. And finally, of course, got to throw in the curveball of what happened in this last year. And honestly, this idea of decentralized trials is not new to the pandemic, but it really welcomed it in. We all used Zoom before. We had e-meetings. We knew how to use technology, but we didn't do it 8 hours a day from our homes because we didn't have to. The same now is true of driving clinical research into a completely virtual setting, a completely decentralized setting. This idea that you don't have to go to a site or to a doctor's office. So the operational implementation of a pragmatic trial is now turning much more towards this idea of a decentralized trial. One of the areas that I think is the most exciting about this is the creativity around medical devices, ingestibles, and wearables. How we actually, some patients might enter a clinical trial and they get a package in the mail with instructions on how to take a medicine and something that they take that's literally a metric collecting data, or they are sent a Fitbit and they wear that watch and it's tracking information and sending it in, streaming data. So there's many new mechanisms to how to execute experimental clinical trial therapy research. And I think this is one of the most interesting areas right now as we consider the same components of pragmatic trials. We're bringing around real big world data for real and big drugs, but now we have to think of doing that virtually. And that's got some really good statistical methodology considerations, especially as we consider streaming data. And a concept that everybody gets excited about is this idea of efficacy in real time. So how can we actually not only stream data, but stream insights, know immediately the status or the health of our clinical trial? So I think there's a lot of opportunity as we consider how design changes and how the methodologies, how we handle that data from those design change in the next few years. So with that, I will wrap up. Hope you guys enjoyed it and looking forward to some of the discussion.
Kelci, thank you very much. This was very interesting information for me. I've been around the area of designed experiments for a lot of years. probably 50. and I've had almost no either exposure to or academic training in clinical trials. And so I've been learning a lot about this over the last year or two. The closest I ever came to really knowing anything about clinical trials was some consulting work that I did for a consumer products company when they made things for skin ointments and that sort of thing. And there are regulatory aspects surrounding that, and so they did have a statistician that handled that, because they did have to do clinical trials to make sure that this lotion that you were putting on your skin wasn't going to cause your skin to fall off or have some really terrible adverse effect. And she actually ended up doing her PhD with me. She decided that it was time to move on to a PhD, but she did her dissertation on something completely different. So I learned a little bit about those types of clinical trials from her. But I find all of this really fascinating, because this seems to be an area where the design and the data management and the analysis are all moving very rapidly. Lots of new things going on, and I'm just astounded.
Yeah. It's a very fast moving field now, and to your point though, it's been almost all in the last 5 to 10 years.
Yeah.
It really was that traditional, simple, randomized block design, don't make any changes, was the way you went. So clinical trial design used to be thought of as super, super simple. And now, of course, we have all these complexities that are fun, but good to handle. So I think more and more people will start getting into the design aspect of clinical trials, because now it's posing some really interesting problems.
I agree with you, and I hope that's the case, because I think there are a lot of really interesting methodological considerations from not only as you point out, the analysis and data management side, but also on the design side. And I can remember 4 or 5 years ago, actually getting a book on clinical trials, and I said, well, I'm going to read this book, and I'm going to become knowledgeable about clinical trials. And after reading about the first half of the book, I said, well, what's the big deal? There's nothing new or exciting, or I already know all this. Well, obviously that's not the case today. So I find this topic to be really quite fascinating, and I really appreciate your taking your valuable time to spend with us to talk about these issues. Do you have any references or anything that you could provide for us that would be good background reading or good introductory material that would get people started? Because we don't.
Sure, yeah, and I should have added that. I can send that along.
If you would, that would be great, because we don't talk about this at all in the course, and my textbook does not say one word about clinical trials, because, as I say, I don't know much. So if my theory in the book is if I don't really feel like I'm an expert on this, I'm not putting it in the book. So that would be really helpful, and I think we can arrange to share that with all of the people in the course without any difficulty. So that would be just great. We can probably figure out how to make that available along with the recording from this session. So if you would do that, would be really helpful. I think your talk is going to stimulate a lot of interest on people in learning more about this. Okay, let's maybe talk about some of the questions. We do have a few that have come in ahead of time, and I got a quick look at this one before we started, but let me go ahead and read this question, because it is a really good question. And this person says they see the value of designed experiments, and how can we use them in the following situation. So I'm going to read this, and then we'll talk about the answer. In a continuous process, oil water separation, water treatment plant, as you know, there are many variables, especially chemical doses, to optimize with current settings established after years of trial and error. Once operations people become comfortable with existing plant setting, engineers may not be given much latitude on the size of moves that can be made to those established settings to build a statistical model for fear of upsetting production. However, if the moves permitted are too small, then as you have explained, noise in the process and process measurements may preclude building a good model. One of the things I learned early on when I was a young engineering student and working for a division of Union Carbide, my boss, the plant manager, told me, he said, Doug, if you want to find out what happens in this process when you change a factor, you actually have to change the factor. That is, you have to change it over a big enough range to be able to see something. Well, what approach then? The question goes on to say, what approach should be taken if the engineers and testers are not given much latitude on the range over which settings can be adjusted during testing so that noise doesn't prevent drawing good conclusions? Can it be the case that DOE simply cannot be applied if operations only permits small changes? That's the biggest challenge he sees at the moment, and he'd like to have some advice on this. Well, this is actually a fairly common problem in a lot of production settings. Because making large changes can produce a lot of bad product or maybe cause safety issues. And there is a technique that can be applied very successfully in this situation called evolutionary operations. And it goes by an abbreviation EVOP. And there is some material on EVOP in my textbook, and there's a lot of other material out there. There's actually a couple of books on EVOP. And the idea of EVOP is that we do set up a factorial design, but we make relatively small changes in the factor levels, but we run it continuously. We run it over and over and over and over again. And so we get lots and lots of data at each of the test conditions. And as we know, as the sample size increases, your ability to detect changes, even small changes in the response variable, increases greatly. And so the idea of EVOP is that you will set up this factorial experiment around your current operating conditions, and you'll run it for a period of time until you get an indication that maybe we need to shift our current operating conditions to maybe one of the other design points or to some location in between the design points. And then we'll reestablish another phase of EVOP, as they call it, and we'll do it again. And this is a very, very useful technique in a lot of chemical and process industries, because there you are in situations where you just simply can't make big changes. And I highly recommend that the individual that sent in this question look at the material that's out there on EVOP and figure out whether or not this fits the bill in solving his problem. And you know, Tom, I think you're the person that sent this in. Please feel free to contact me if you need some help in tracking down some of these resources to help you get started on EVOP. And the nice thing about EVOP is it is based around the idea of a simple factorial experiment. So all the EVOP analysis that you need to do can be done with standard software like JUMP. So it would be pretty easy to actually implement this. Great question.
Tom had another great one he shared in the chat just now. I'd love to read out an answer too. He said, apologize if this is off topic, but why did AstraZeneca and J&J not detect possible COVID vaccine blood clotting problems? through the safety trials. So I know this is a sensitive subject, and I am by no means, I will say right now, I'm not on any of the data monitoring committees for any of these clinical trials. I do actually know some of the statisticians who are, but it's a great question, especially as it relates to design. So why didn't we see this, right? And it goes back to the traditional design aspects around, inclusion exclusion criteria and how we set up who can enter a clinical trial. It also goes into the fact that the blood clot, I think it was occurring at a rate of like 0.00001, like 10 in 100,000 vaccines where this blood clot is now occurring, and talk about power to detect those types of differences. But it really comes back to this idea that inclusion exclusion criteria is how we set up clinical trial designs. Typically, because of good clinical practice, we want to think of treating somewhat healthier people. There's also many of the blood clots were in women. And it's a very well-known fact right now. A huge area where we need improvement in clinical trials is diversity in the populations. But that's also hard because women might be pregnant or could get pregnant. But if you don't enroll pregnant women, how are you going to know if a drug is going to help them? And then if they want to take it, what is the information they should go on? So I think both of these companies have been doing very well with regard to the transparency of their data, but there are just clear constraints to clinical trials. One of the reasons real world data and this idea of allowing more diverse populations to be a part of clinical research is necessary. I hope that answers that question.
I think that's a good answer, and it certainly expands on what my theory was. My theory was that this only happens a few times out of a million subjects. And what sort of sample size would you have to be able to detect that? You just don't have a large enough sample size to be able to get that kind of information reliably. You'd have to have millions of people in your trial to be able to, and it's not possible.
Yeah. Well, and it is the prevalent highlighting example right now, but there are many drugs that have much greater toxicity effects or adverse event effects that just really aren't found because back to that diversity of the types of populations and the protocol deviations or inclusion exclusion criteria that are structured sometimes very far in advance during the creation of the clinical compound, not allowed to change as the trial progresses.
Okay, here is another question that comes out of the course on factorial and fractional factorial designs. And let me read this. Is it possible to have additional treatments in split plot designs as extra control factors? For example, of 3 to the 2, 3 squared, plus 1 factorial experiment in a split plot. And yes, you can. It will probably not be a standard textbook design because that extra factor could have possibly multiple levels, not just 3. It might have 2 or 4 or 5 levels. Can you incorporate this in a split plot structure? Yes, you can. But constructing that design is probably, I think, going to be an optimal design. It's not going to be a standard textbook design. So if we're using JUMP, and by the way, he mentioned SAS or R in the question. I don't use SAS very much because I don't like to program things. So I use JUMP because I'm a creature of pull down menus. And I don't use R at all for a variety of reasons. And Kelci, you might even comment on this. I'm not sure that regulatory agencies are comfortable with people using R because it's open source software. And there are a lot of people that even if they don't work in regulated industries, they go out to work for a company and the company will not let them use R because they don't want open source software on company machines. And so when I teach my experimental design course, you know, you know, we don't do R, you know, and if people come to the class and they say, well, I'm from the stats department and I'm taking the course and we use R, and I say, well, okay, just keep it over there because we're not using R over here in engineering. So you're going to use JMP, but JMP has a lovely custom design platform, and you can, I'm sure, create the kind of design that you're talking about here without any real difficulty, because all you need to do is if for example, if your control factors are your hard to change factors, then all you have to do is indicate that that is a hard to change factor in the custom platform, and it will create the split plot design for you. And once JMP knows that you have a split plot design, it automatically does the correct analysis. It automatically deals with the two error sources, the whole plot and the split plot error sources that are required to do the statistical analysis. So the JMP system is very smart. It has information built into the design table that it knows exactly what you did when you set up that design, and then it guides the analysis down the appropriate path. So I think this is easy to do. I don't know. I think this is how I would attack that problem.
No question. I even though I've changed companies, JMP is unparalleled, especially with respect to DOE. I love it. I would use it. I haven't bought it yet. I need to. All time, of course, one of the most intuitive and user-friendly softwares. Your question earlier is, it's actually a really interesting and relevant one right now, around open source software at the FDA or at regulatory agencies. It's a huge consideration because one of the key parts of the clinical trial process is complete accuracy, reproducibility, and traceability of the statistical analyses. And traditionally that has always meant SAS, but now as more and more systems are working towards validating open source softwares or including, so many people are moving just more to this idea of you don't pick one, you can use many languages. In fact, that's what the FDA does. I'm not, of course, going to speak for them, but they've publicly announced their use of all of those technologies.
Okay.
To my knowledge, I don't think there's been a end-to-end submission in only an open source software like R, but I know those considerations of how you incorporate that are in the works, but it comes back down to what's true with any software. If it's off rogue on a desktop, how do you control the environment and know what that environment is? And there's aspects of that. If you are going to use R, there's costs. No matter what you do, there's costs when it comes to software, whether it's in complexity, validation, accuracy, or use. So it's a great question, but it actually is an area where they're moving. Clinical statistical programmers and data scientists are really trying to adopt a multi-language approach to solving disease. In fact, in the COVID trials, in the development of Moderna, it was the first example in the submission. They used a custom R package for part of the clinical trial design for their adaptive data collection. I forget what it was called, but I can look that up and send it to you. So, you know, use them all, right? Less exclusion criteria. That's the more real data and real programs.
It is a question that I wonder about, because I do consulting work with other types of companies, not necessarily pharmaceutical companies, but people that make commercial products, and they are very reluctant to have any kind of open source software on their computer systems because of security. And
right.
You know, I totally understand that, because you know, having your proprietary research data hacked because you were using some kind of open source software would be a pretty disastrous kind of situation for a lot of people. So
in fact, what you're saying is even more relevant. The way pharma rarely lets you have anything on your laptop, because what if your laptop gets stolen and it has clinical data on it? Everything is locked down. So regardless of whether it's open source or commercial, this concept of a managed environment that is not local is traditionally where pharma is moving,
Yeah.
which leads to a lot of complexities in cost and accessibility. So how can we use the machines we have, but make sure that the data and the programs are secure? That's a common concern in clinical trials.
Same thing in the financial services industry.
Yeah.
If you work for a bank or a financial services company, your laptop doesn't have the ability to plug in, thumb drives or USB drives or to download data onto an external device.
That's true in regulatory too, as well. Yeah, I can't. I've multiple times I would go to visit the FDA, and we decided it was easier to do it on Zoom because if I was there, I can't get on their network and I can't plug anything into their machine. So it was like, you have me in person, but we can't work together. So
yeah, I knew people that worked for banks. In fact, a couple of my former students worked for banks, and even though they could work from home for most of what they did, if they actually had to get into customer databases, they actually had to go to a bank site where they had a secure internet connection that would enable them to look at customer financial records. But they couldn't download any of that onto their computer. They had to do it from a secure facility at the bank. Same sort of idea.
yeah.
Okay. So
another question came in on the chat. I can't promise I got a good answer to this one, but it's a great question.
Can you see this one?
Yeah, early on in the rollout, AstraZeneca COVID vaccine was criticized for lack of research data on some of the age groups. Is that a failure of their experimental design? Why did they fail to collect those data? I can't say I know. I don't know why or what their clinical design was, except for that they it's a different mechanism. Of course, that's the vector viral, not the mRNA technique. And from my knowledge, I think that was early, and many times that is a concept of an adaptive trial, especially if you are doing benefit risk analysis, is you start with the group where it's likely to be most successful and then show some measure of efficacy. And it's back to this idea of how you start up with a homogeneous group and then expand that to the diverse groups. I'm not sure why they did it that way, but I don't think that I would consider it a failure of their design, because now as that was an early phase of here's the group it can support. And as we gain more and more evidence, I think that's actually the epitome of an adaptive trial, to show how you start with something solid, know it can work for something, and then expand out. But of course, they took a much more structured research approach to the development of that vaccine, because of course it was after the immediate emergency use vaccines that went through. I think it's also related, of course, the emergency use vaccines were put out and specifically helped to target the older population where COVID was such a problem. And many of those older populations took that. And so there could have been a consideration. I have no clue. This is just a thought that they started with the age group where they saw the most benefit of producing the next vaccine that could be available or accessible to people. But I don't see it as a failure of their design. I just think of it as the evolution of how they executed that design.
That's exactly what I'm thinking too. And again, I'm not an expert on this, but they probably started with the population that they could get easy access to, and where they thought that they would have hopefully some success, and then you expand it as things go on. I have a friend that suffered from colon cancer. This is some years ago. And his oncologist wanted to enroll him in a clinical trial, and that clinical trial was being managed, I believe, by the Cleveland Clinic. And he thought that he would be a really good candidate for this trial because of age and family history, and some other characteristics of the particular cancer tumor that he had. And that essentially directed him to a particular arm of the trial, and I'm a particular treatment regime. And I suspect that the kind of trial he was in was the sort of thing that you're talking about here, where as they move through the trial, they start trying to reach out to include different types of patients in the trial to see how they perform. And by the way, his treatment was totally successful. So he doesn't have you're not suffering from colon cancer anymore.
Good.
Okay,
That's
we
awesome.
have one more question, it looks like, that has come in. What methods should we use for analysis of 2 to the k factorial designs when outcome is not continuous nor binary, but rather non-parametric? And as an example, they give average revenue per customer or average order value. Well, if these are in fact averages, you can appeal to a central limit theorem here and use the standard analysis of variance for normal theory data. That would probably not be a bad place to start. There is data, however, that comes along that truly is not continuous, but it might be subjective. Rank data, for example. And in fact, a lot of non-normal data can be analyzed by simply converting the non-normal response data to ranks. For the single factor completely randomized design, there is a technique called the Kruskal-Wallis method that is used for the analysis of ranks. And that method is discussed in my textbook. It turns out that if you look at the Kruskal-Wallis statistic carefully, that it is a monotone function of the F statistic, the usual ANOVA F statistic. And so it has been observed by other researchers that applying the ordinary analysis of variance to rank data works really, really well. And so what I would suggest that you do is think about converting your 2 to the k data to rank data, and then use a standard analysis on ranks. I could give you an example from my own personal experience, okay? Back in the middle 1980s, when I was at the University of Washington, I've got interested in joining up with a group of people in starting a winery operation. And so we had a group of 17 people that we bought 24 acres of vineyard land and started trying to figure out how to make Pinot Noir. Well, none of us knew how to make Pinot Noir, but we knew how to run designed experiments. And so we started doing 2 to the k minus p fractional factorial designs. We did 8 factors, 16 run designs, 2 to the 8 minus 4 every year for 4 years. And so at the end of every year's production, we'd make the wine, and then we would do taste test. We would get a panel of people together and we would do taste test. And so each taste tester would rank the 16 samples, 1 being the best, 2 being the next best, 3 being the next best, all the way on down. Number 16 was the worst. And we had some really bad stuff early on, believe me, because we didn't know what we were doing. But we ended up using standard methods for the analysis of 2 to the k designs on that ranked data. And it worked really, really well, because over a period of 4 years, we developed a very good commercial winemaking process for Pinot Noir. And we took our first commercial release in 1990, and we submitted that to the American Wine Competition and won with 2,000 wines, and we won a gold medal. So we figured it out. And by the way, the data from one of those experiments is in chapter 8. It's one of the examples in chapter 8, or I'm sorry, one of the exercises in chapter 8. So you can go find that one of our 16 run fractional factorial experiments. All the rank data is there, and you can take a look at that. So ranks work great. All right, we're at the end, and we're just about out of time, as it turns out. We are taking a summer break on the Fireside chats. So there is no Fireside chat in June and July. We will be back in August. The date is August 18, and I'm looking forward to teeing this up again and having a lot of fun with you folks. And I want to thank Kelci so much for taking her time to spend with us and preparing such a great presentation. And thank you again so much for coming. And I hope everybody had as good a time and learned as much as I did.
Thank you, Doug. It was my pleasure. This was great.
Bye-bye.